BIN 310 Microbial Genomics. Let's have a short look at what we are doing this autumn. First, these 4 lines of text here. The world has changed. I see it in the water, I feel it in the earth, I smell it in the air. Now, do you recognize these lines of text? Because these are actually the first lines in The Lord of the Rings by Tolkien. And actually, I think if you look at the movies, it also starts out with these 4 lines. Why on earth do I mention this here? Well, I think this is an excellent sort of phrase for describing the microbial genomics. The world has changed. We sequence it in the water, we sequence it in the earth, and we sequence it in the air. Look at this picture here, taken from a scientific paper describing this. We sequence it in the earth, in the air, in the water, and even inside ourselves. So this is modern microbial genomics. We sequence the microbes or the prokaryotes from all kinds of environments, and indeed the world has changed. Because this has happened over the last decade or so. 2 decades if we are liberal. The planet of the microbes, yeah. So why are the prokaryotes or the microbes so important to study? Well, first, a Hampel cord. You know what a Hampel cord is? It's around 40 meters long from one end to the other. Imagine now 1 centimeter on the Hampel cord is 1 million years. Then we can fit the whole area, the whole era of life on Earth on this, going from left to right? So here we are today. And then the microbes have been around all the time. Life on Earth is like 3,800 million years old. At least that's our current estimate. And from the very first time, this was microbes or bacteria-like organisms. Of course, they were not looking the same as they are here today. These are the bacteria. At this time, we can now go out and sequence the bacteria, but they are relatives, right? It's the same sort of type of organism that has been around all the time. So the eukaryotes, they came in around here, like 15 meters ago, you could say. And so we can see that for a long time, for the majority of the time, the life on Earth, the prokaryotes were alone. There were no other or type of organisms. And then the eukaryotes came along. The fishes that we maybe think of as sort of simple organisms or have been here for always, no, they have only been around for like 5-6 meters. And the land plants even shorter because life began in the oceans, right? The land plants 4 or 5 of these meters, and the insects only like 3-4 meters. So, I mean, there is a colleague of mine here at NMBU wrote a book called The Planet of the Insects. Well, not really. The insects have only been here for a short time compared to the microbes. And the humans, we think of as the most important species. The last 2 millimeters. Our history is super duper short here. Unimportant in a sense. If you look at the historic perspective, of course, at currently there, I couldn't say that humans are unimportant for planet Earth, but that's another story. So the microbes are everywhere, have been here for always, and you find them in deep sea sediments, in the clouds, and in and on all the other organisms, because they have had a lot of time, right, to develop and fit into all niches on this planet. And here you can see a tree of life. Yeah. And the reason I show you this tree of life, so you can see there are many branches here, right? Because this tells us something about the diversity, right? How different are the different organisms. This green branch down here are all the eukaryotes. And in this branch, Ophistochonta, you find the humans, far down. And this is not super big compared to this branch here, which is the archaea, which is much bigger. These are prokaryotes, single-celled organisms. And here you have the bacteria, the other single-celled organism. So these two together are the prokaryotes. And you can see that there's enormous diversity between them. And you can see there's a huge leap here between bacteria and archaea. They are actually quite different, much more different than archaea are from us, actually. And that is, of course, because they have had plenty of time to evolve to such a richness in genomes. The black part here, or the sort of the blackish branches, that's what we call biological dark matter. And these are the new sort of organisms that we have discovered recently. Because in the old days, you could say, we could only study this part of biology by isolating and growing them in the lab. And only a small fraction will grow in the lab. So most of them live out in the environment, and we couldn't study them until now. So that's how the world has changed. Now we can sequence them everywhere, right? So these black branches here are the ones we just recently discovered. Just recently. We didn't know about this 20 years ago. I saw this paper here saying that based on sort of what we have up until 2020, we estimate that what we know here is approximately 2% of the global prokaryotic taxa. So there are we are still lacking sort of 98% of the branches in this tree. That's what they say. So there is a lot of dark matter, biological dark matter, still to find out there. And we find new every day. This grows. So this tree is probably outdated already. But compare it to the small branch of eukaryotes, right? We don't discover that many new eukaryotes these days, right? It could be in the deep sea oceans or in the Amazon jungle if there is something left of it. But otherwise we know pretty much all there is. But here we know close to nothing. Okay, metagenomics is the reason for this discovery. So let's say a few words about it. First of all, it's based on the DNA sequencing technologies, right? And that's why this has happened over the last 20 years. And we will talk about lots about sequencing and sequencing data in this course. Not always about metagenomics though. But metagenomics, you collect all the DNA from some environment, could be all kinds of environments you can see here. Then you isolate the DNA from all the organisms in there without sort of sorting them or identifying them, and you just sequence it. And then you do the sorting and identification down here. And this is the bioinformatics part, right? So this is the wet lab part that we will never see in this course. This is outside the scope of this course. This is where we are in this course, after the sequencing machine. You should learn about this in other courses, how to prepare DNA and so on. There are typically two types of sequencing when we talk about metagenomics, and we will come back to this towards the end of the course actually. The marker sequencing, where you just sequence the target gene from all the organisms in the environment, and you compare them by how this gene look like. And then you have the shotgun sequencing, which means you sequence all DNA. And you can say that in the beginning of this course we will not talk about metagenomics at all. We will talk about isolates, where we have a single organism, and then we also do this sort of shotgun, but I mean we only have one organism, so we don't typically call it shotgun, but it's the same sequencing technology. You sequence all DNA from the environment or from some single organism. So what we see then when we have done some sequencing is this, reads. And this is regardless of what type of sequencing we do really. We end up with lots of short DNA sequences representing a piece either of a gene or of a genome. And this is a complete mess if you don't use bioinformatics to sort things out here. And we can have a lot of these reads in a data set. Hundreds of thousands, millions, and even hundreds of millions reads in a single data set, single sample, right? So this can be horrible. And we need some heavy computers and stuff to do this, as we will see. We will not work on the biggest data sets in this course, of course. That would be silly because we are like 60 students and we can't really, that will take enormous resources. So we will use some rather small data sets, but the methods will be the same. Let me say a few words about relevance of microbial genomics to activities at NMBU. Of course, first of all, in basic microbiology, this is important, right? Being able to sequence the genomic content of the microbes is really important for understanding the microbes themselves, for their own sake. In human and veterinary medicine, it's also important, and we have all these activities at NMBU. Yeah, and this is important because I mean there are some pathogens among the bacteria that make us ill, right? Or the animals are ill, but also for our digestion system and our general well-being, you could say. Our interplay with the microbes has been recognized as being important. Food production and food safety. Yeah, this is all about food production. You use microbes in the production process, in dairy products and so on, but also food safety, right? Again, the pathogens. We don't want to have the bad bacteria in our food. And again, genomics is it's important for some aspects of this, right? Of course, the interactions between production fish, animals, or plants and their microbiota. That's also quite a lot of research on that. And I mean, NMBU, traditionally, the production animals or plants or fishes have always been important here. And genetics and genomics for these organisms is also really important. But the interplay between their genetics, you could say, and their microbes has been much more focused on the recent years. Environmental studies, yeah. In this setting, we often use the microbial composition as an indicator of the environmental status, right? If there are something wrong with the environment, then you will find that the microbial composition changes. Not the amount of microbes, but which microbes are flourishing, you could say. So this is a typical use of microbial genomics in this setting. And even forensics. I mean, it's not something you think of with respect to NMBU perhaps, but we also have some activity in this direction. And microbes, you think forensics or crime scene in investigation, it's all about the human DNA, but it's not. It's also about the bacterial DNA that you find on a crime scene. It can be used for important information. So microbial genomics is really important in many aspects, and probably some I haven't mentioned here at all because I don't know or I forgot. So it's a nice thing to know a little bit about this. It's not unlikely you will come across problems involving it. in other fields as well. So the selected topics in microbial genomics is the title of this course, and I have to say, with respect to the course itself, the world has changed as well. Because if I look back like 10 or 12 years here to 2011, this course was called Models and Algorithms in Bioinformatics. And it was sort of more theoretical, where we looked into the sort of ideas of the various tools that were used in bioinformatics back then. And some of them we use still. What has changed is that this year, or actually in the last few years, I have changed the course content and the way we approach things completely. I now focus much more on the practical use of software tools. Now, originally this course was made for those who take a master in bioinformatics. And then you need to know a little bit about how things are, how the tools are working, right? If you're going to sort of make tools, you of course need to know how they are working. It's like if you're going to make a car, you need to understand how cars work. However, most of the people coming to this course are not going to build tools. They are going to use tools. And that's a completely different story. You can still use a car without knowing all the details about how it works. And that's my sort of focus now. Use the tools rather than dig into them. And this is more than just because of the students coming to the course have a different sort of need. It's also to do with my pedagogical philosophy, which has changed. Because I now think that even if you're going to learn how the tools work, you should still start out by use the tool first, and then you can dig into how it works. So it makes sense to start here anyway. That's my conclusion after being both a student myself and being a teacher now for many years. I think that this is the best way of learning. Do the practical part first, then the more theoretical part. When I was a student, this was clearly opposite. When I was a student, we came to a lecture where a professor was talking about the theory, and we didn't really understand. Well, we understood the theory perhaps, but not really why we should understand the theory. And then afterwards, we went to some exercises and solved some problems, right? So that was reversed order. So we focus on the tools for the more basic data processing. And that's also a little bit important here, because the raw data we start out with are from the sequencing machines, and that's the type of data we will start with here now in this course. And we will only look at the more basic processing of this, the basic processing steps, and we cover wide instead of digging deep. So we will visit sort of a number of sort of processing this in a number of directions, but we will not dig into the biological questions very much. So we will sort of stop at the very sort of first steps. And the reason for this is that if we were digging in one specific direction here, the course will not be relevant for others than those who are interested in this direction. So by stopping at the sort of more basic processing steps and instead look at many different versions of them, I think it becomes more relevant to a wider audience, you could say. But it perhaps less interesting in the sense that the really interesting biological questions we will not dig into here. But that's something you have to do yourself in a master project or whatever, PhD project perhaps. Then you can dig into your questions, and then you should of course also go further and use other tools outside what you see in this course. This is only the sort of basics. Yeah, I like to think of programming as having some resemblance to playing music. I don't really play music, so I'm not sure if this is a good it's a good metaphor, but uh, at least when you're playing music, you first have to learn the keys on the keyboard, and then you can start combining them into playing music. And I think this is the same with programming. First, you have to sort of understand the coding elements, and once this is sort of obvious to you, then you can start making programs. You start thinking in terms of these coding elements, right? And then you can sort of start making music, programs that sort of flow. And I think that this pipeline science, as I call it, this microbial genomics, the tool-based microbial genomics, is somewhat resemblance sort of meta programming, because instead of programming in the classical way, we now have a long range of tools, right? We have a toolbox with many tools in. I mean, should know how each tool work, what it can do and what it cannot do, the input and output, right? What can we use the various tools for? And then we can start combining them into solving almost any problem, right? And that's pipelines, because we then typically make some sort of pipeline, or we use tools in a sequence, right? So that we can come from the raw data and to solving the problem that we are actually looking for. But this takes some time, right? You will not become a musician after this course, but at least you will learn some basic tools and a little bit how to use them, but only by a lot of exercising here you can become the musician. So you need to know the keys, the software tools. You will at least hear about many of them here, or some of them here. You also need to do some coding to link them together, right? Some Unix shell coding we will see a little bit here in this course. We are not supposed to know, I will not require any previous knowledge, and we will only learn a little bit here at this course. But this is important for sort of tying it together. And the other the more classical programming tools like R and Python. I use R. I can read Python, but I'd never use it. You should use both. I mean, younger generation, you should know both of these. But in this course, we will focus on R. At least I will. Everything I put out and all you can ask me about is R. You shouldn't ask me to help you debugging Python programs. You can make them if you like. I will not deny you that, but then you are on your own here. But there are plenty of others you can ask, of course. We will look into quite a number of different tools in this course. So hopefully you will have some there that you can put in your sort of toolbox that you expand later.